So let's continue where we ended in the previous class. We are talking about the experiment that Thomas Hunt Morgan had performed, and we know that Thomas Hunt Morgan is the father of experimental genetics. Gregor Mendel is the father of genetics. Thomas Hunt Morgan just came and he did confirm the experiment which was performed by Sutton and Boveri. So first he got a white-eyed fruit fly. This is Drosophila melanogaster. He crossed it with a red-eyed fruit fly. Now the white-eyed, this is the normal one. And then the red-eyed is mutant. And what he saw after mating these two is that all the offsprings were red-eyed. And there were four offsprings. So this is known as the F1. Because this is a physical appearance, this is known as the phenotype. It's not the genetic makeup. That's what you can see, right? Physical phenotype. And what he did is now he got one of these offsprings. He got a male offspring, which was red-eyed, and a female offspring that was red-eyed. And what he saw is that there were three offsprings from these two offsprings that were red-eyed, and then one that was white-eyed. And so from there, he made a conclusion. Okay, he made a conclusion. So, firstly, let's talk about the experiment that was done. Remember what we are saying, what was crossed here. So, what was crossed is he got one offspring and crossed with another, and that's what gave us the results. So if you see, we have got what we call the phenotypic ratio, right? Phenotype is a physical appearance of an organism. So if you look at the phenotypic ratio, can I write P for phenotypic? So I'm going to write P and ratio. Let me just finish the word. So phenotypic ratio. So what was the phenotypic ratio here? Look at how many red do you have? Red-eyed fruit flies. There are three, right? So that is a three. And how many white-eyed do you have? Only one. So this is the ratio that he discovered, 3 to 1. But there is also, before you can have a physical appearance, you actually have the genetic makeup. And that's known as a genotype. So the genotypic ratio that he discovered was actually 1 to 2 to 1. Now we are going to understand these ratios more a bit later. So what I just want to mention now is that these experiments which were done by Thomas Hunt Morgan using Drosophila melanogaster was just to confirm that the work which was done by Sutton and Boveri in their chromosomal theory was actually true. And that as well suggested that Mendel's experiment was correct. Okay. But what Mendel did is that when Thomas Hunt Morgan later on used the Drosophila melanogaster, but Mendel got plants. When he got a lot of plants and then he started crossbreeding them, he started to see some of them when you got the red ones and the white ones. Later on, there were different colors that were coming up. That was Mendel's experiment using a plant. However, Thomas and Morgan used the fruit fly, Drosophila melanogaster. Okay, so what should we know from this experiment? This experiment is just confirming the chromosomal theory of inheritance, and Remember what we talked about the chromosomal theory of inheritance, that chromosomes come in pairs and that the pairs separate in meiosis, and that the different chromosomes will assort independently. We understand the law of independent assortment when we start talking about the Mendelian laws. Now, let's talk about a chromosome itself. What really is a chromosome? Though we mentioned it a bit in the previous class. How is a chromosome made? Right? Now, a chromosome is made of chromatin. Okay? So, chromosome is made of chromatin. The next question should be, what is chromatin made of? Or what is chromatin? All right? This chromatin is basically DNA plus histones. So chromatin is DNA plus proteins. And those proteins, we call them histones. Okay, so there, that is what we have to know. So how do you get to form then this chromatin? What is going to happen is that the DNA is going to be wrapping around. You know what makes up DNA, right? DNA is basically nucleic acid. And what's a basic unit of nucleic acid? What is a basic unit? So again, first you need to understand when we are talking about these histones, how many histones are there in DNA? Just a moment. Okay. So when it comes to histones which are in DNA, you have got eight types. Eight. These, remember, are proteins. So I've got eight histones that are going to be found in DNA. And these are basically proteins where the DNA will be able to wrap. All right. So let's now understand how a chromosome is going to be made up. It all starts like this. You have got, remember, DNA is a double stranded molecule, right? So you have got this double stranded molecule like this double stranded molecule. So what is going to happen? This is our DNA, which is a double stranded molecule. We call it a double helix. And what is going to happen is that this DNA is going to start to condense and condense. I'm just making it smaller like that. Just think of it like this is where you are, and then it's going away from you, so it's hard to see far, right? Yeah. So this DNA, when it becomes longer and longer, it will start coiling around these proteins, which we call histones. So we're going to see we have got an histone here, and then the DNA here is going to coil. Let me write another color so that it can make sense there. So I'll put an histone here. I'll put another histone here. Okay, like that. So this, you are going to see it is going to come and coil around the histone, and then this one also come and coil around the histone, and they are going to continue coiling around the histone just like that, right? So we are calling these proteins here as histones. So when the histone is when the DNA helix is called to the histone, what do you form there? You are forming chromatin. So this is going to become chromatin and is much more condensed. Okay. So of course you have got histones there, but I'm not just showing the histones. The histones are there. And then later on, what you are going to see is that this structure will now start wrapping around itself. Will start wrapping around itself. Let me write it well. So you see the structure starts to condense even all the more. Starts to condense even all the more. It's condensing even all the more. Condensing even all the more. Just like that. And then it is condensing about itself. Continue condensing about itself. You see how what it becomes. So that's how you are going to end up with this structure. And what are we calling this structure? This is what we call a chromosome. So that is how a chromosome is made. You are having a double helix coiling around histones, and that is going to give you chromatin. Okay, and then you continue to coil again like that. That is to condense. The DNA will continue to condense and condense and condense, and then you form a chromosome. Okay, hope that makes sense. Now, when you have got, there is something that we call a nucleosome that is going to be formed. So as you are combining, because remember, you have got, of course, the DNA that is wrapped around the histones. Okay. So at this point where the DNA is getting to wrap around the histones like that, we call that as a nucleosome. Okay. A nucleosome. And then the nucleosomes, they'll continue like that, coming to condense and condense and condense, and then we are having to form a chromosome. Okay. So basically, that is that. And this chromosome that we are seeing here is basically made up of, because just this structure, you combining an histone and DNA, that's a chromatin. Chromatin. Please, not chromatid, but chromatin. Okay. So that is important there to know. So in 1900, chromosomes were identified as colored bodies in the nucleus that are capable of getting to divide before cell division. So the structure of a chromosome in human, it is going to look like this one that we can see here. So we have got 23 pairs of chromosomes. That is a first basis. That's how we can classify chromosomes. We can classify them on the pairs. We have got 23 pairs of these chromosomes. Now, when you look at a chromosome again, a chromosome has got two parts. So this is going to be a chromosome that I'm writing. So imagine we have, anyway, let me not draw it like a flower. I can just make it simple. So we can have a structure like this. Okay, so here is our structure. So we are saying that we have got 23 pairs of chromosomes. That is the first basis of how we can classify chromosomes. When you are classifying chromosomes, chromosomes have got two arms. You have got the P arm and the Q arm. Okay. So let's say here we have got this a chromosome, and then B also is a chromosome. So A here is a single chromosome. can see it's just alone. And B was a single chromosome, but it made a copy of itself. So this is a duplicated copy. It has got a duplicated copy, meaning it is exactly the way the other one is. Okay. And then when you look at the structure, you have got two arms. You have got the longer arm and the smaller arm. So the smaller arm is called the P arm. This one is smaller. And then the larger arm is known as a Q arm. When you look at the duplicated chromosome, they are both attached on the middle here, and we call that as a centromere. Okay. So the centromere is a position of DNA which acts as a link between these two guys. Now these two guys, they are known as sisters, and they are known as sister chromatids. So this one alone is a chromatid. This one also is a chromatid. Because they were formed by duplication, we call them sisters. So sister chromatids. That is also important for us to understand there, right? So remember, I've mentioned of chromatid, but earlier on I mentioned a chromatin. So what is the difference between chromatin and chromatid? So that is one thing that we should be able to differentiate. So when you are talking about, of course, so there are three terms you should be able to differentiate: chromatin, chromatid, and also a chromosome. When you're talking about chromatin, it is just a substance found in the cell nucleus that has got DNA and protein. So that substance made up of DNA and eight histones, right? Eight types of histones. That's what we call a chromatin. So when the cell gets to divide during cell division, chromatin is going to condense. Okay. So this whole structure here was the chromatin. This chromatin is going to condense and then it is going to form chromosome. So chromosome is just a condensation of chromatin. Now, what happens when the chromatin condenses to form chromosome? It is then going to split into two identical strands. Okay, so the chromatin, you can see, condensed, and then it forms the chromosome. This chromosome now can split into identical strands, one this side, one this side, and those are called chromatids. So chromatin is the indistinguishable mass of DNA molecule, whereas chromatids are a part of a chromosomes attached to it with a centromere. Hope we've gotten that. So chromatid is basically a part of a chromosome, and those two parts which are separated by centromere, while chromatin is the actual DNA plus histone that to make up the chromosome after condensing. Okay, so I wanted us to understand that. So there are different types of chromosomes depending with the location of the centromere. So let's write some. You can have a structure like this. So I'm going to write four types, and I want you to identify what you can see as a difference amongst them. So what can you identify as a difference amongst these chromosomes that I've drawn? You can see for the first chromosome, the centromere is on the middle. The Q arm and the P arm are equal. This type of chromosome is known as a metacentric. Okay, metacentric chromosome. And then if you look at this one, the centromere is not on the middle. One part is at least further from the other. This is known as a submetacentric. And then if you look at this one, the shorter arm, which is the P arm, is nearly disappeared, is nearly not there. This is known as an acrocentric acrocentric chromosome. And then you have got a chromosome which does not have a shorter arm. Okay, it just has one arm, meaning that the centromere is actually at the end of that chromosome. That's known as telocentric. So there. Now, when we come to the human karyotype, so we are talking about human karyotype. What do we mean by karyotype? So a karyotype is basically a representation of the number of chromosomes. So in a human, a representation of the number of chromosomes doesn't have any telocentric. This one is not in human, okay, but these others can be there. Karyotype is just one way of representing all the chromosomes in a cell. And each cell in our body, except the gametes, have got 23 pairs. Now, the gametes don't have 23 pairs. Take note of that. All right. So, the other type of classification that we can use to classify the chromosomes, apart from the location of the centromere, is whether the chromosome belongs to the sex cells or they belong to body cells. Okay. So, sex chromosomes are known as allosomes. You know the X chromosomes, we have got X and Y. These chromosomes are the ones which determine if a person is going to be male or female. While the other chromosomes, okay, just belong, they are not sex chromosomes, other, which belong just to the body, to other body parts. So, what do we call those which are non sex chromosomes? So, non sex chromosomes are known as autosomes. Okay, so that's what we need to know. Now, when it comes to the sex chromosomes, we have got X and Y. So, for females have got only X chromosomes, and then males are going to have both X and Y. And the X chromosome is actually longer than the Y chromosome. So, this is how they are. So, again, a chromosome is an entire chain of DNA along with a group of stabilizing protein. We already mentioned that. So all cells in the body will contain two sets of chromosomes except a sex cell. And in human somatic cells just means body cells. So in any body cell, in any somatic cell, we have got 23 pairs. Okay, somatic, also known as body cells, have got 23 pairs. So if it is 23 pairs, how many then do we have? 23 times 2, that's 46 chromosomes. And these 46 chromosomes, what do we call that? That's known as a diploid. haploid number, also known as 2N. Okay, N represents 23 chromosomes. And then when we go to the sex cells, okay, the sex cells have got an haploid number. Sex cells have got 23 chromosomes. You can see this is not pairs, this is 23 chromosomes, and this is an haploid number. Haploid number is just N. You only have 23 chromosomes. Somatic cells, you have got 46 chromosomes. That is because for these sex cells, you, after coming together, which ones are the sex cells? Okay, actually, not even the sex cells, but the gametes. What are the gametes that you know? So, gametes are basically sperms or spermatogonia and the ovum. That is because if the ovum comes together with the sperm, then you are going to form now something that has got 23, right? Pairs now. It's now a pair because you are getting from your mother and also from your dad. Otherwise, the sex cells are going to have only 23. No, yeah, are going to only have 23 chromosomes. The gametes only have 23 chromosomes. Okay, hope this statement is that clear and we have understood. So this is very, very good for us to understand. Okay. So another thing that we need to know is then, now that we understand what a chromosome is, we should know what a gene is. Okay. Because when we go, let's say we pick a chromosome. Let me try to write a chromosome. Here is a chromosome. I'm just writing a single one that is doesn't have a sister chromatid, and there is a centromere. So when you come on this chromosome, you are going to find a specific location, okay, that is going to contain a gene. So let's say, for example, on this location is telling you you need to have blue eyes. Blue eyes. This is a locus or loci if you're talking about a lot. Locus. So locus is basically a specific location on a chromosome that contains a gene for a specific trait. For example, you should be short. That location is just telling you about that trait, either height or eye color or air type or whatsoever protein that is in your body. The information is going to be found on a specific locus. Okay. Now, what is really, because we are saying that locus, what have we said the locus is? A locus is a specific location on a chromosome on a chromosome, and this specific location on a chromosome contains a gene, okay, for a specific trait. Don't forget it is a specific trait. Like for example, eye color, maybe your also your body color, anything. But then what is a gene? Because we are saying that a gene is going to be found on a locus and it will tell us the kind of trait we are going to show. So a gene is a segment of the DNA that codes for a protein that is used to express a trait. Okay, it's a portion. A gene is a specific portion, or say, let me say segment. Okay, segment of DNA that codes. By now we know what it means if we say code, right? Because we've talked about translation, the formation of protein. So that codes for a protein that is used that is used to express that trait. Okay. Let me now not put that trait. Let me put a trait. Because if I put that, I'm of course referring to the statement on top. So segment of DNA that codes for a protein that is used to express a trait. For example, on this segment, on this position, locus is just a location. But on that location, you have got a gene. And that gene is telling you you need to make a protein that will make your eyes to be blue. So that is what we call a gene. But then the thing is this. In diploid organisms, chromosomes always come in pairs. Okay, in any homologous pair, one chromosome comes from the father and the other chromosome comes from the mother. So meaning that the the information that we have, the reason as to why I am the way I am is the information that came from my mother came and connected with the information that came from my father, right? And that is how I ended up being the way I am. But then if my mother is light and my father is not light, why is it that I'm not light? That has to do with which gene from either the mother or the father is much stronger. Now, for you to form a gene, you need information from both the mother and the father, as you have said, right? So now let's understand that. So let's say this is the chromosome coming from the mother. This chromosome is coming from the mother. And then here is a chromosome that is coming from the father. There. So what you are going to see is that if on this position from my mother we have got information talking about my, let's say, skin color, maybe from my mother information coming is that I should be light, but on the same location information coming from my dad is that I should be dark. So even when you are talking about different things, right? Light and dark, you are talking about the same thing. You are talking about skin color. It's just that what color, okay? What color, but it is all skin color. Let's say on this position, if from my father, or this one is from my mother, if it's from the father, let's say you should have, let's say, black eyes, and then brown eyes, sorry, and then from my father it is you should have blue eyes. So about the color of the eyes, the information is found on the same position, both from the mother and from the father. But then it can not always be talking about the same thing. Of course, the information from my mother can be light, my father is also light. So maybe the information from both can be saying you should have light skin from both the mother and the father, meaning that definitely I'm going to be light skinned because both information is coming from both parents. But then if my father's trait is saying you should be black, my mother's trait is saying you should be light, the one which is stronger amongst between the two is what I'm going to be, and that is known as the dominant trait. So this specific position, the information that is coming from the father and from the mother that I'm inheriting, we have got what we call alleles. Okay, so we have got a pair of alleles. Okay, so when two alleles come together, they will now form a gene. So now, what is an allele? That is what we need to understand. And what is the difference between an allele and a gene? So an allele is a different form of the same gene. Okay, different form of the same gene. Remember this gene that we are talking about, let's say it's a gene for eye color. But then the eye color is not always brown. It can also be blue. It can also be another color. So the different forms of that same gene that we are talking about is what we call an allele. Hope we have gotten it. Okay. So if they say what is an allele, different form of a gene. Okay. So gene, what is a gene? A gene. So now when you're comparing a gene and an allele, a gene is going to specify a trait. For example, the eye color, or the height, or the size. But an allele will tell you what size, what color, what this, what this. So that's what an allele answers. The allele answers what? The gene is just telling you the trait. Okay, the feet, the shape of the feet. That is the gene that goes for the shape of the feet. But then the other will come. What is the shape? So you need now the actual shape. Okay, maybe from the father is that the feet should be big, from the mother is the feet should be small. So the one which is dominant is what you are going to have. Hope we have gotten that. We have gotten the differences between an allele and a gene. Okay, so when you're talking about chromosome, because you are getting a chromosome from your mother and a chromosome from your father, the chromosomes that you inherit should be homologous. So the chromosomes we inherit should be homologous chromosomes. But then what does it mean? What is an homologous chromosome? Or what are homologous chromosomes? So for a chromosome to qualify to be homologous, number one, they should have the same size. Okay, the same size and the structure. Number two, they should carry genetic information about the same type of physical trait. So that genetic information is a gene, right? So they should carry genes about the same trait. And then, for example, if a chromosome contains a gene for eye color, its homologous chromosome will also contain a corresponding gene that is carrying eye color. And such pairs of genes that code for that, a pair of gene that codes for the same trait, we call them alleles, as we have said. So you need to note that because homologous chromosomes have got similar size and shape, each allele pair are found at a similar location, and that similar location is known as a loci. So these guys are supposed to have the same shape, right, and the same size. So there is no way you are going to call these as homologous chromosomes. If let's say this is talking about eye color from my mother, but then from my father the locus for eye color is down here. No, that can't happen. They should be at the same location. Okay, hope we have understood. So what is the other thing that we should know? Even though each allele pair contains similar genes, the genes do not have to code for identical traits. We already mentioned that. For example, from your mother, you can say, okay, you are supposed to be blue-eyed. From the other parent, you're supposed to be brown-eyed. But it's the same gene, but then it's not the same trait that you are coding for. So by definition, that homologous chromosomes should have the same size. Let's look at sex chromosomes. For the sex chromosome, what you are going to see is that the X chromosome is something like this. So this is the X chromosome, and then this is the Y chromosome. So do you think these two chromosomes are homologous or not? So these chromosomes are not homologous. So X and Y chromosomes, even if both are coding for sex, they are not homologous chromosomes because they don't have the same size. Okay? So that is what we need to know. This is it about the introduction. Let's do some questions. Okay, but before I even go to these questions, let's understand some things. So, just one more concept. Since we have talked about a chromosome, okay, we have talked about the shape of a chromosome. When it comes to a chromosome, look at how it is. It's like this. This is a chromosome. But this chromosome that you can see here, these two guys, this one and this one, what do we call them? These are sister chromatids. Okay, these are sister chromatids. Sister chromatids there. Now, for these two guys, which are sister chromatids, what happens is that during cell division, there these cells will come. These chromosomes are going to be pairing up. These are sisters. They are not homologous chromosomes. They are sisters. Okay. So sisters are made by duplication. One had to duplicate and it formed another one. So during cell division, what will happen is that there's going to be another pair of sister chromatids. So actually this guy is homologous to this guy. But then they just made the copy of, each one made the copy of itself, so that during cell division, they are going to separate. Each cell, after it gets such a chromosome, is going to be the same as the one that has remained. So you can see that you have got four chromatids. Because we have got four chromatids, of course, we'll talk about this during cell division. We call this as a tetrad. Tetrad just means a pair of two homologous chromosomes. But because we call it tetrad because we refer to the chromatids, there are four. It's also known as a bivalent. Okay. Also known as bivalent. Why bivalent? Because we have got two chromosomes, one, two. So bivalent is referring to the chromosomes themselves, but tetrad is referring to the chromatid. Hope we have gotten that. So this basically happens during cell division. Okay. So a bivalent is made up of two chromatids. Is that true? Sister chromatids, four homologous chromatids. One and two above are correct. One and three are correct. Which one do you think is the correct answer? Okay. So let's start with A. Two chromatids. Is it made up of two chromatids? That's not true, right? It's made up of sister chromatids. Is that true? Partially true, but all right, it's partially true. But it's not all of them are sisters. Like these two are sisters, these two are sisters. But if you get this and this, they're not sisters. Okay. Four homologous chromatids. This would be qualifying. Okay. Four homologous chromatids. Also, or you can say two homologous chromosomes. So that's the answer. Two homologous chromosomes or four homologous chromatids. Which of the following is part of eukaryotic chromosome in any phase? Now, these questions will supposed to answer it. Anyway, let's answer. In any phase, whether during cell division or not, which one is part of a eukaryotic chromosome? The centrioles will only appear after when cell division is happening, but histones are part of a chromosome. Okay. Nucleosome, this structure, the nucleosome, we had mentioned anyway. Let's mention the definition. By definition, because I'd mentioned what makes up a nucleosome, but then if it is by definition, what is a nucleosome? So a nucleosome is a basic structural unit of DNA packaging in eukaryotes. Okay, it's basically made up of DNA, which is wounded around 8 histone proteins. That's what we call a nucleosome. So when you want to form a chromosome, the nucleosome is going to fold in themselves and then condense. So this is of course part, because histones are always part of DNA. And I mean, histones are always part. Like, you can't have a chromosomes without histones at any point. And you can't have a chromosome without nucleosome. Chromatin is a basic unit when you combine the histones, right? And the and the DNA. Centromere connects the DNA, isn't it? It connects the two chromosomes. So it's always part. But the centriole will only appear when cell division is happening. Otherwise, what happens is that you barely see this on a microscope. You barely see this DNA, the chromosome properly. Like, you barely see it unless it condenses. Okay. So which of the following is not part of a eukaryotic chromosome in any phase? The centriole is not part of the chromosome. Centriole is just going to be an appearance that is going to be seen during cell division. We are going to understand when we talk about cell division. Chromosomal theory of inheritance states that genes occupy specific positions on chromosome. This is true, right? Homologous chromosomes segregate from each other during meiosis. Okay, this is true. Segregation. Non-homologous chromosomes are sorted independently during meiosis. This is also true. So the answer is 1, 2, and 3 above are correct. An animal somatic cell contains 12 chromosomes, while a nucleus of sperm cell contains only 6 chromosomes. What does N equal for this species? Now look at this. An animal somatic cell. So the somatic cell contains 12. We had mentioned that somatic cells contain a diploid number. So this is actually 2n. So meaning that 2n is equal to 12. So what is n? What is going to be found? Just divide. That's 6. Right? So that's the haploid. Describe chromatin. The other name for chromatid. Is that true? No. A repeating unit of a chromosome. Is that true? Okay, let me put a dot first. An alternative form of a chromatid. Is that true? No. A thin thread of nucleic acid plus protein. Is that true? Yes. Sequence of nucleotide base which code for a complete polypeptide. No. So chromatin is basically uh, nucleic acid, which is DNA, around histones. Choose a statement which is false. Eukaryotic chromosomes are made of DNA and protein. Is that true? This is true. It's not false. In the chromosome, the basic protein are called histone. This is also true. In the chromosome, the acidic protein are called non-histone phosphate. Let me put a dot here because I didn't talk about that. The basic unit of association between DNA and protein is called a nucleosome. Is that true between DNA and DNA and protein? This is true, right? You remember where I circled? We said that when you have got a nucleosome and then you have got the DNA pi rounding it like that. I circled it and said that's a nucleosome. Okay, that's true. And then for the next one, none of the above statement is false. Now look at 3. In the chromosome, the acidic protein are called non-histone protein. Okay, just a second on this one. All right. So the non, where is that? The non-histone proteins are indeed acidic in nature. So this is also correct. So the answer is none of the statement is false. Okay, linkage prevents homozygous condition, prevents independent segregation, promotes recombination, prevents heterozygous condition 2 and 3. Now, of course, we are going to talk about this in the next class. When you have got homologous chromosomes like this, what happens is that they are going to form a link. Can you see this link here? So this link is there to promote recombination, genetic recombination. Okay, so that's what it's there for, but we'll discuss in the next class. If BB is a gene pair of an individual, then allele for this gene are, so the alleles are basically these same guys. So B is an allele and B is an allele. For example, this is coming from my mother, you should have black skin. This is coming, the small one, let's say, is coming from my father, you should have light skin. So we differentiate the alleles by using capital and small, just to mean if you use capital, it's just telling you which one is dominant, which one you are going to say. So if capital B is for black color, then that is what I'm going to be. I'll be black. Okay. So the alleles for this gene pair, it's this one, small b and capital B. The basic structure unit of a chromosome is the, okay, this one I'll allow you to answer. I'll give you 30 seconds. All right. So the basic structural unity of a chromosome, is it a centromere? No. Telomere? No. Kinetomere? No. Centrosome? No. Nucleosome? That would be the thing. Remember, nucleosome is basically histones, and then you have got a nucleic acid wounded, uh, wrapped around it. Which of the following is an example of a discontinuous variation? And I should have mentioned about continuous and discontinuous variation. Let me just talk about them a bit. Okay. So when you're talking about continuous and discontinuous variation, these are basically traits that you inherit, right? That you can get from your parents. So when you talk about, I'm going to write here, let me put continuous here. and then discontinuous. We actually are able to measure these guys. We can write a graph for the continuous variation and the discontinuous variation. That's how we can tell. So I want you to interpret what you can see. So on continuous variation, you are going to see a graph like this. On discontinuous variation, you are going to see something like this. So what can you see? So for a continuous variation, these are traits which have got no limit to the value. For example, the height. Someone can be 2 meters, someone 2.1, another one 2.2, another one 2.01, another one 2.001. So you can, there is no limit to what you can have. So examples of these discontinuous variations include the weight. You can have someone who is very fat, someone who is very thin, someone between, someone like that. Weight, height, even your heart rate. Okay, someone's heart rate can be 110, another one's 60, another one 40, another one, it can be very high, very low, or anything. Even your length or your size. Okay, whatsoever that you can talk about. But when it comes to discontinuous variation, these are distinct categories and there is no in between. For example, blood group. You are either blood group A, blood group B, blood group AB, or blood group O. You cannot be blood group O and A at the same time. No, you can only have one. Besides blood group, you also have fingerprint. You only have one fingerprint. You can't have a lot. That is a discontinuous trait. That's why the graph cannot be like this one. Also, eye color. So it's either you have got brown or blue. You can't have brown and blue at the same time. No. Also, tongue rolling. So you can try to do it. You can try to fold your tongue. Try to fold it as though you are taking it towards the throat. Not everyone can do that. Okay? Only some people can roll their tongue. So which of the following is an example of discontinuous variation? Height. This is continuous. Okay, weight and height are continuous. Human intelligence. Intelligence is also a continuous variation. Someone can be dull, someone very intelligent, another one very, very intelligent, another one very, very, very intelligent, right? Okay. And then color blindness. Okay. So for color blindness, what can you say about color blindness? Do you think you can have between or none? Or you can have between, maybe you are a bit blind, you are very blind, or you are not blind at all. Or it's either you are color blind or not color blind. 30 seconds. Okay. So color blindness is a discontinuous variation. Okay. Color blindness is a discontinuous variation. So it's either you are color blind or you are not color blind. There is nothing at no, I'm just a little color blind. Okay, so which of the following is an example of discontinuous? That's color blindness. So that is it where we end on this class. Thank you so much. In the next class, we talk about cell division.